Assalamu alaikum dear students. I welcome you all in my next zoology class dedicated for 12th standard. In this class we are dealing actually with human reproduction, and up to this stage we have completed the basic things. For example, what actually the scrotum means, and we have studied the morphology of testicles, as well as we have studied the anatomical structures of testes and the one of the important and specific role of testes, that is testosterone formation, and we also studied testosterone metabolism. Today the main topic will be, let us suppose this is a testicle. Attached with the testicle is a special structure here. This special structure we call epididymis, and from this epididymis a special tube arises. This special tube is known as vas deferens. So today's topic will be focused on these two structures. Attached with the testicle is a special structure. It's the testicle. Attached with the testicle is a special structure that you call epididymis. Along with epididymis, we have to study vas deferens. These two structures. Easy to understand? Just what I require is your kind attention. Let we start our today's topic. Let us suppose this is scrotum, and this is the partition of the scrotum, muscular partition, and inside the scrotum, let us assume this structure be the testicle. Obviously here also there will be a testicle. Now attached with this testicle, let us suppose this is the anterior end. Obviously this will be the posterior end. At the posterior end of each testicle there is attached a comma shaped structure like this. Attached with the testicle there is a comma shaped structure. If I will draw this comma shaped structure here, this comma shaped structure actually contains highly coiled tube like this. This structure that you call epididymis. This epididymis is a comma shaped, highly coiled tubular structure that is usually 4 to 6 centimeter long. 4 to 6 centimeter long. However, if this coiled structure is to be unfolded, if this comma shaped structure or if this coiled tube is to be extended, it can extend up to 6 meters. The primary function of this tube, is storage and transportation of sperms. The primary function of this tube is simply storage plus transportation of sperms. Storage and transportation of sperms. These sperms are actually formed from the seminiferous tubules of the testicles that you already know. We have studied it in our previous lectures. Now, the first and the foremost thing after introduction is, let we move to the wall of this seminiferous tubule. Let us wall of this epididymis. This is epididymis, highly coiled tube. The wall of epididymis is actually made of pseudo-citratified epithelium. Wall is made of pseudo-citratified epithelium. In class 11th, you have studied two important things. The first concept that you have studied is simple epithelium. The second concept that you have studied is compound epithelium. Their teacher tells you that simple epithelium is made of just one layer of cells, and compound epithelium contains multiple layers of cells, or that is called citratified epithelium. Citratified epithelium or compound epithelium means tissue or membrane made of several layers of cells. But here, as far as epididymis, wall of epididymis is concerned, this wall of epididymis is made of pseudo-citratified epithelium. When I say pseudo-citratified epithelium, the diagram for pseudo-citratified epithelium looks like this. Pseudo-citratified epithelium is actually made of single layer of cells. Pseudo-citratified epithelium is that epithelium that is made of single layer of cells. But this epithelium appears to be multi-layered. This epithelium appears, this membrane or this epithelium appears to be multi-layered due to the presence of two different types of cells. One is the tall cells that you see on the diagram, and second is the presence of short cells. This epithelium appears multi-layered due to the presence of two different types of cells, like this. One is the tall cell, second is the short cell. This epithelium is actually known as pseudo-citratified epithelium. You have studied it in class 11th. You can check the 11th textbook and study more details about pseudo-citratified epithelium. Right now, my concern is that this comma-shaped structure attached with the testicle called epididymis is actually having a wall that is made of pseudo-citratified epithelium. This pseudo-citratified epithelium of epididymis is responsible for production of nutrients, responsible for the survival of superbus, or responsible for the maturation of superbus. What I said is that epididymis produces wall of epididymis. This wall of epididymis produces secretions. So these secretions are rich in sodium. These secretions are rich in potassium. These secretions are rich in glycoproteins. Glycoproteins. As well as the wall of epididymis that you call pseudo-citratified epithelium. It has another function that is known as absorptive function. It has another function. That function is known as absorptive function. That is the wall of this epididymis. It is this epididymis. Let us assume this is a testicle. From this testicle, superbus are synthesized, superbus are produced, sperm cells are formed. These sperm cells will then enter into this coiled tube. This coiled tube is known as epididymis. What I said is that wall of epididymis is known as pseudo-citratified epithelium. It has absorptive function. When I say it has absorptive function, I mean it is this wall of epididymis that absorbs extra fluids present with sperm cells. along with sperm cells, the additional fluids, if any, present in these sperm cells are actually absorbed in the wall of epididymis. Now, next point. Sir, wall of epididymis also produces a special fluid. That special fluid is known as phosphoryl glycerol choline. This is also used as a sperm nourishment. This is also used as nutrient fluid or nutrient for sperm cells. What I told is that epididymis is actually a comma-shaped, highly coiled tubular structure attached with the posterior side of each testicle. Now, this wall of epididymis is made of pseudo-citratified epithelium that has several functions. The first and the foremost function is that this epithelium, wall of pseudo-citratified epithelium of epididymis produces nutrients that are essential for survival of sperms. These nutrients, these fluids produced by epididymis contain sodium ions, contain potassium ions, contain glycoproteins. They also contain phosphoryl glycerol choline that is also used as nutrient by the sperm cells. Simultaneously, what I said is next point that the primary role of epididymis, this comma-shaped structure, is storage and transport of sperms. Simultaneously, the third function that I told is that it is this epididymis, the wall of which is made of pseudo-citratified epithelium, and this wall has absorptive function. It absorbs the additional fluids present with sperm cells. The next point that you should understand is that epididymis also produces inhibitory factors. Epididymis also produces inhibitory factors or inhibitory peptides. Let me make you people one thing clear first of all. Let us suppose this is the testicle. This testicle will produce something called sperm cells. Each day, millions of sperm cells are formed. Each day, millions of sperm cells are formed. Millions of sperm cells are formed in the adult testicles. If these sperm cells are to be extracted, from the testicles, these sperm cells lack motility function. What I mean to say is that these sperm cells, actually the sperm cells move by whiplash movements. First of all, sperm cells move by, in our further lectures we will tell you that initially sperm cells move by zigzag movements. Then sperm cells show whiplash movements. When I say whiplash movements, I mean sperm cells have a special tail structure. The tail is used by the sperm cells to move towards, actually the sperm is responsible for fusion with the egg. Sperm has a role to fuse with the egg in the female reproductive system because humans have internal fertilization. So in the female reproductive system, sperm has to swim up to the egg. This swimming power is achieved by whiplash movement as well as by zigzag movement. Actually, what happens is that sperm cell has a tail-like structure. When we will study sperm in detail, there I will tell you, it is this sperm that shows movement. But what I told is that if the sperm cells are to be removed from the testicle, the fresh sperms lack movement. They are non-motile. They can't move. They are non-motile. However, When sperms are formed in the testicles, they then enter into this epididymis. In the epididymis, sperms stay usually for 14 days. But in your textbooks, it may be written that sperm cells reside in the epididymis. Sperm cells are stored in the epididymis for 15 to 20 days. In these 15 to 20 days, or usually in these 14 days, sperm cells gain the power of movement. Sperm cells gain the power of movement. When the sperm cells gain the power of movement, that too in the epididymis, that concept is written in our books. That is known as physiological maturation of sperms. Physiological maturation of sperms. When I say physiological maturation of sperms, I mean sperm cells gain a power to show movements. However, there is no complete maturation. Make this point clear that there is no complete maturation of sperm cells. There is no complete maturation of sperm cells in the human male reproductive system. When we study another factor that is called capacitation, in female reproductive system we have to study something called capacitation, sperm maturation. There I will tell you what is the complete maturation, what does complete sperm maturation mean. But right now you should understand this very basic concept that sperm cells develop partial maturation. That is, sperm cells show movement after they stay in the epididymis for 15 or 20 odd days. This maturation that I call physiological maturation of sperms. Simultaneously, right now I told you that wall of epididymis produces inhibitory peptides. It is these inhibitory peptides that prevent final maturation, of sperm cells. Final maturation of sperm cells occurs in female reproductive system. Here only basics, physiological maturation, slight movement of sperm cells. Sperm cells gain the power of movement in the epididymis. But the complete maturation of sperm cells, the concept called capacitation, occurs in female reproductive system. What I told is that it is the wall of epididymis that produces inhibitory peptides. These inhibitory peptides produced by this wall of epididymis prevent the final maturation of sperm cells. Now, next point that you should understand is that this epididymis actually contains three parts. The first part is called head epididymis. Second is called body, and the third part is known as tail. What I told is that attached with the testicle is present epididymis. This epididymis contains three important regions. The first region being the head, second body, third tail. Head is also known as caput, C-A-P-U-T, caput epididymis. Body is known as corpus epididymis. Tail is known as cauda epididymis. So, caput epididymis, second part is known as corpus epididymis, third part or tail part is known as cauda epididymis. It is from this tail part a dilated tube arises. This dilated tube is known as vas deferens, but that will be discussed in next lecture. Right now, have a quick recapitulation. You may not get 100% of the factors because when we study completely male reproductive system and female reproductive system, there I will tell you something called sperm cells. There I will tell you the sperm maturation. There I will discuss with you people the capacitation, the acrosome reaction. But right now the basic thing is that testicle has a special structure attached at the posterior end. That structure you call epididymis. This epididymis is actually made of highly coiled tubular structure. The wall of this tube is made of pseudo-citratified epithelium. This pseudo-citratified epithelium produces nutrient fluids rich in sodium, rich in potassium, rich in glycoproteins. Simultaneously, this epididymis produces inhibitory factors that prevent the complete maturation of sperm cells. The third point is that it is this epididymis, the main role of which is storage of sperm cells for 15 to 20 days. The fourth function is that sperm cells forward in testicles lack the power of movement. However, their setae in the sperm cells, the setae of sperm cells in the epididymis makes them little bit motile. They gain the power of movement. This partial maturation, partial means slight maturation of sperm cells is known as physiological maturation. This physiological maturation occurs in epididymis. The fifth point that I told is that it is this epididymis that basically contains three parts. The first part is known as head, that you call caput. The second part is known as body, that you call cauda. That you call corpus. The third part you call tail, that you call cauda epididymis. What I told is that this epididymis attached with testicle contain three regions. Head, divisible into three parts: head, body, tail. Head is known as caput epididymis. Body is known as corpus epididymis, corpus. And the tail is known as, this tail region is known as cauda epididymis. Hope you got the basics clear. If you don't understand it, repeatedly watch it and you will get the concepts. Do like, share and subscribe my channel.